Good morning, everyone. Today I'm going to discuss the genetic mechanisms and evolutionary processes and selected topics in biogeography and distribution. The natures present scientists with a puzzle. Humans share the earth with millions of other kinds of organisms of every imaginable shape, size, and habitat. This variety of living things is called biodiversity. How did all these different organisms arise? How are they related? These questions make up the puzzle of life's diversity. What scientific explanation can account for the diversity of life? The answer is a collection of scientific facts, observations, and hypotheses, which is known as evolutionary theory. Evolution, or change over time, is the process by which modern organisms have descended from ancient organisms, while a scientific theory is a well-supported, testable explanation of phenomena that have occurred in the natural world. As Darwin developed his theory of evolution, he worked under a serious handicap. He didn't know how heredity worked. Although Mendel's work on inheritance and peace was published during Darwin's lifetime, its importance was not recognized for decades. This lack of knowledge left two big gaps in Darwin's thinking. First, he had no idea how heritable traits pass from one generation to the next. Second, although variation in heritable traits was central to Darwin's theory, he had no idea how that variation appeared. Evolutionary biologists connected Mendel's work to Darwin's during the 1930s. By then, biologists understood that genes control heritable traits. They soon realized that changes in genes produce heritable variation on which natural selection can operate. Genes became the focus of new hypotheses and experiments aimed at understanding evolutionary chains. Another revolution in evolutionary thought began with Waitson and Crick's studies on DNA. Their model of the DNA molecule helped evolutionary biologists because it demonstrated the molecular nature of mutation and genetic variation. Today, molecular techniques are used to test hypotheses about how heritable variation appears and how natural selection operates on that variation. We now know that many genes have at least two forms, or alleles. Animals such as horses, dogs, and mice often have several alleles for traits such as body size or coat color. Plants such as peas often have several alleles for flower color. All organisms have additional genetic variation that is invisible because it involves small differences in biochemical processes. In addition, an individual organism is heterozygous for many genes. An insect may be heterozygous for as many as 15% of its genes. Individual fishes, reptiles, and mammals are typically heterozygous for between 4 and 8% of their genes. Evolution in genetic terms is any change in the relative frequency of alleles in a population. For example, it is responsible for both the remarkable similarities we see across all life and the amazing diversity of that life. But exactly how does it work? Fundamental to the process is genetic variation, upon which selective forces can act in order for evolution to occur. This research work examines the genetic mechanisms of evolutionary processes and selected topics in biogeography and distribution, specifically on: first, the descent and the genetic differences that is heritable and passed on to the next generation. Then, mutation, migration, genetic drift, gene shuffling, and natural selection as genetics mechanisms of evolutionary processes, and then the importance and sources of genetic variation. We will also study what is Hardy-Weinberg principle, how biogeography considers species geographical locations, and how species distribution reveal evolutionary events. We've defined evolution as descent with modification from a common ancestor, but exactly what has been modified. Evolution only occurs when there is a change in gene frequency within a population over time. These genetic differences are heritable and can be passed on to the next generation, which is what really matters in evolution: long-term change. Compare these two examples of change in beetle populations. Which one is an example of evolution? Beetles on a diet or beetles of different colors? Imagine a year or two of drought in which there are few plants that these beetles can eat. All these beetles have the same chances of survival and reproduction, but because of food restrictions, the beetles in the population are a little smaller than the preceding generation of beetles. Most of the beetles in the population, say 90%, have the genes for bright green coloration, and a few of them, 10%, have a gene that makes them browner. Some numbers of generations later, things have changed. Brown beetles are more common than they used to be and make up 70% of the population. The difference in weight in example 1 came about because of environmental influences that low food supply, not because of a change in the frequency genes. Therefore, example 1 is not evolution, because the small body size in this population was not genetically determined. This generation of small-bodied beetles will produce beetles that will grow to normal size if they have a normal food supply. The changing color in example number 2 is definitely evolution. These two generations of the same population are genetically different, but how did it happen? A mutation could cause parents with genes for bright green coloration to have offspring with a gene for brown coloration. That would make genes for brown coloration more frequent in the population than they were before the mutation. To understand more about this, let us discuss the genetics mechanisms such as mutation, migration, genetic drift, gene shuffling, and natural selection as genetics mechanisms of evolutionary processes. Mutation, which is also the fuels of evolution. A mutation is a change in a nucleotide sequence in DNA. For example, a T nucleotide could undergo a mutation and be replaced with an A nucleotide. Mutation from one allele to another obviously can change the proportion of particular alleles in a population, but mutation rates are generally too low to significantly alter Hardy-Weinberg proportions of common alleles. Many genes mutate 1 to 10 times per 100,000 cells divisions. Some of these mutations are harmful, while others neutral or even rarer and beneficial. Also, the mutations must affect the DNA of the germ cells, or the mutation will not be passed on to the offspring. The mutation is so slow that few populations are around long enough to accumulate significant numbers of mutations. However, no matter how rare, mutation is the ultimate source of genetic variation in a population. Next, the gene shuffling, or the non-random mating, which concentrate alleles locally. In this case, individuals with certain genotypes sometimes mate with one another, either more or less frequently than would be expected on a random basis. This phenomenon known as non-random mating. One type of non-random mating is inbreeding, or mating with relatives, such as in the self-fertilization of a flower. Inbreeding increases the proportions of individuals that are homozygous because no individuals mate with any genotype but their own. As a result, inbred population contain more homozygous individuals than the predicted by the Hardy-Weinberg rule. This is why populations of self-fertilizing plants contain so many more homozygous individuals than outcrossing plants, which have higher proportion of heterozygous individuals. Non-random mating alters genotype frequencies, but not allele frequencies. The alleles are just distributed differently among offspring. Say for example, you do not look exactly like your biological parents. Even though they provided you with all your genes, you probably look even less like any brothers or sisters you may have. Yet, no matter how you feel about your relatives, mutant genes are not primarily what make them so different from you. Most heritable differences are due to gene shuffling that occurs during the production of gametes. Recall that each chromosome of a homologous pair moves independently during meiosis. And as a result, the 23 pairs of chromosomes found in humans can produce 8.4 million different combinations of genes. Another process is crossing over, also occurs during meiosis. Crossing over further increases the number of different genotypes that can appear in offspring. Remember that a genotype is an organism's genetic makeup. When alleles are combined during sexual reproduction, they can produce dramatically different phenotypes. Thus, sexual reproduction is a major source of variation within many populations. Sexual reproduction can produce many different phenotypes, but it does not change the relative frequency of alleles in a population. Next in line is migration, which moves alleles between populations. Migration is defined in genetic terms as the movement of individuals between, into, or out of a population. It can be a powerful source upsetting the genetic stability of natural populations. Migration includes movement of individuals out of a population called immigration, or the movement of individuals out of a population called immigration. If the characteristics of the newly arrived individuals differ from those already there, and if the newly arrived individuals adapt to survive in the new area and mate successfully, then the genetic composition of the receiving population may be altered. Departing members of a population take their alleles with them. Likewise, new members entering and interbreeding with an existing population may add new alleles. But migration does not require the movement of the entire individuals. Wind carries a plant's pollen for miles, for example, spreading one individual's alleles to a new population. The fourth genetic mechanism is a genetic drift, which occurs by chance. Remember that genetics is controlled by the laws of probability. These laws can be used to predict the overall result of genetic process in a large population. However, the smaller a population is, the further the result may be from what the laws of probability predict. This kind of random chance in allele frequency is called genetic drift. But how does genetic drift take place? In small populations, individuals that carry a particular allele may leave more descendants than other individuals, just by chance. Over time, a series of chance occurrences of this type can cause an allele to become common in a population. Genetic drift may occur when a small group of individuals colonizes a new habitat. These individuals may carry alleles in different relative frequencies than did the larger population from which they came. If so, the population that they found will be genetically different from the parent population. Here, however, the cause is not natural selection, but simply chance, specifically the chance that particular alleles were in one or more of the founding individuals. A situation in which allele frequencies change as a result of the migration of a small subgroup of a population is known as founder effect. One of the examples of the founder effect is the evolution of several hundred species of fruit flies found on different Hawaiian islands. All of those species descended from the same original mainland population. Those species in different habitat on different islands now have allele frequencies that are different from those of the original species. Genetic drift may result also from a population bottleneck, which occurs when a population size drops rapidly over a short period. This bottleneck randomly eliminates many alleles that were present in the larger ancestor population. Even if the remaining individuals mate and restore the population's numbers, the loss of genetic diversity is permanent. Cheetah, for example, are currently undergoing a population bottleneck. Until 10,000 years ago, these cats were common in many areas. Today, just two isolated populations live in South and East Africa, numbering only a few thousand animals. Inbreeding has made the South African cheetahs so genetically alike that even unrelated animals can accept skin grafts from each other. Researchers attribute this genetic uniformity to at least two bottlenecks, one that occurred at the end of the most recent ice age when habitat changed drastically, and another when humans slaughtered many cheetahs during the 1800s. Note that the loss of alleles from the cheetah population has been random. It is therefore different from natural selection, which weeds out alleles that reduce fitness. The last one is the natural selection, which molds evolution. Imagine that green beetles are easier for birds to spot, so brown beetles are a little more likely to survive to produce offspring. They pass their genes for brown coloration onto their offspring, so the next generation, brown beetles are more common than in the previous generation. As Darwin pointed out, some individuals leave behind more progeny than others, and the likelihood they will do so is affected by their inherited characteristics. The result of this process is called selection and was familiar even in Darwin's day to breeders of domestic animals. In so-called artificial selection, the breeder selects for the desired characteristics. For example, mating larger animals with each other produces offspring that are larger. In natural selection, Darwin suggested that environment plays this role, with conditions in nature determining which kind of individuals in a population are the fit or best suited to their environment, and so affecting the proportions of genes among individuals of future populations. The environment imposes the conditions that determine the results of selection and thus the direction of evolution. The variation inherent in each species means that some individuals in each population are better than others obtaining nutrients and water, avoiding predators, tolerating temperature changes, attracting mates, reproducing. The heritable traits conferring this advantage are adaptations, which is the feature that provide a selective advantage because they improve an organism's ability to survive and reproduce. The word adaptation in evolutionary sense includes only those structures, behaviors, or physiological processes that are inheritable and that contribute to reproductive success. In any population, individuals with the best adaptations are most likely to reproduce and pass their advantage to their offspring. Because of this differential reproductive success, a population changes over time with the best available. Natural selection requires pre-existing genetic diversity within a population. Ultimately, this diversity arises largely by chance. Nevertheless, it is important to realize that natural selection itself is not a random process. Instead, it selectively eliminates most of the individuals that are least able to compete for resources or cope with the prevailing environment. Let us discuss a natural selection of single-gene traits. These traits can lead to changes in allele frequencies and thus to evolution. Imagine a hypothetical population of lizards are normally brown but experiences mutations that produce red and black forms. What happens to those new alleles? If red lizards are more visible to predators, they might be less likely to survive and reproduce, and the allele for red coloring might not become common. Black lizards, on the other hand, might absorb more sunlight and warm up faster on cold days. If high body temperature allows them to move faster to feed and to avoid predators, they might produce more offspring than brown forms. The allele for black color might then increase in relative frequency. If a color change has no effect on fitness, the allele that produces it would not be under pressure from normal selection. Let us proceed to the natural selection and polygenic traits. When traits are controlled by more than one gene, the effects of natural selection are more complex. The action of multiple alleles on traits, such as height, produces a range of phenotypes that often fit a bell curve. The fitness of individuals close to one another and the curve will not be very different, but fitness can vary a great deal from one end of such a curve to the other. And where fitness varies, natural selection can act. It can affect the distributions of phenotypes in any of three ways: directional selection, stabilizing selections, or disruptive selection. Directional selection occurs when individuals at one end of the curve have higher fitness than individuals in the middle or at the other end. In this example, a population of seed-eating birds experiences directional selection when a food shortage causes the supply of small seeds to run low. Birds with small beaks thus have lower fitness than birds with larger beaks. In the graph, you will see that the dotted line shows the regional distribution of beak sizes. The solid line shows how distribution of beak sizes would change as a result of selection. Stabilizing selection takes place when individuals near the center of the curve have higher fitness than the individuals at either end. This example shows that the human babies born at an average weight are more likely to survive than babies born either much smaller or much larger than average. When individuals at the upper and lower end of the curve have higher fitness than the individuals near the middle, disruptive selection takes place. In this example, average size seeds become less common and the larger and smaller seeds become more common. As a result, the bird population splits into two subgroups, specializing in eating different size seeds. All of those mechanisms I've mentioned can cause changes in the frequencies of genes in populations, and so all of them are mechanisms of evolutionary processes. However, natural selection and genetic drift cannot operate unless there is genetic variation. That is, unless some individuals are genetically different from others. If the population of beetles were 100% green, selection and drift would not have any effect because their genetic makeup could not change. So why is genetic variation important? Without genetic variation, some of the genetic mechanisms of evolutionary processes cannot operate. Genetic variation is studied in population. A population is a group of individuals of the same species that interbreed. Because members of populations interbreed, they share a common group of genes called a gene pool. A gene pool consists of all genes, including all the different alleles that are present in population. The relative frequency of an allele is the number of times that the allele occurs in a gene pool, compared with the number of times other alleles for the same gene occur. Relative frequency often expressed as a percentage. For example, in the mouse population, the relative frequency of the dominant B allele is 40% and the relative frequency of the recessive allele is 60%. The relative frequency of an allele has nothing to do with whether the allele is dominant or recessive. In this particular mouse population, the recessive alleles occurs more frequently than the dominant allele. Gene pools are important to evolutionary theory because evolution involves changes in population over time. In genetic terms, evolution is any change in the relative frequency of alleles in a population. So what are the sources of genetic variation? There are three primary sources of genetic variation. First, mutations or changes in the DNA. A single mutation can have a large effect, but in many cases, evolutionary change is based on the accumulation of many mutations. An organism's DNA affects how it looks, how it behaves, and its physiology, all aspects of its life. So a change in an organism's DNA can cause changes in all aspects of its life. Mutations are random. Mutations can be beneficial, neutral, or harmful for the organism, but mutations do not try to supply what the organism needs. In this respect, mutations are random. Whether a particular mutation happens or not is unrelated to how useful that mutation would be. Not all mutations matter to evolution. Since all cells in our body contain DNA, there are lots of places for mutations to occur. However, not all mutations matter to evolution. Somatic mutations occur in non-reproductive cells and won't be passed on to offspring. For example, the golden color of half of this red delicious apple was caused by a somatic mutation. The seeds of this apple do not carry the mutation. The only mutations that matter to large-scale evolution are those that can be passed on to offspring. These occur in reproductive cells like eggs and sperm and are called germline mutations. Second, gene shuffling that results from sexual reproduction. Sex can introduce new gene combinations into a population and is an important source of genetic variation. You probably know from experience that siblings are not genetically identical to their parents or to each other, except of course for identical twins. That's because when organisms reproduce sexually, some genetic shuffling occurs, bringing together new combinations of genes. For example, you might have bushy eyebrows and a big nose since your mom had genes associated with bushy eyebrows and your dad had genes associated with a big nose. These combinations can be good, bad, or neutral. If your spouse is wild about the bushy eyebrows or big nose combination, you were lucky and hit on a winning combination. This shuffling is important for evolution because it can introduce new combinations of genes every generation. However, it can also break up good combinations of genes. Third is gene flow, also called migration. This is any movement of individuals and the genetic material they carry from one population to another. Gene flow includes lots of different kinds of events such as pollen being blown to a new destination or people moving to new cities or countries. If gene versions are carried to a population where those gene versions previously did not exist, gene flow can be a very important source of genetic variation. Now let us discuss the Hardy-Weinberg principle. What is this all about? To clarify how evolutionary changes operate, scientists often find it helpful to determine what happens when no changes takes place. So biologists ask, are there any conditions under which evolution will not occur? Or is there any way to recognize when that is the case? The answers to those questions are provided by the Hardy-Weinberg principle. This is named after two researchers who independently proposed it in 1908, and it states that allele frequencies in population will remain constant unless one or more factors cause those frequencies to change. The situation in which allele frequencies remain constant is called genetic equilibrium. If the allele frequencies do not change, the population will not evolve. In some populations, these conditions may be met or nearly met for long periods of time. If, however, the conditions are not met, the genetic equilibrium will be disrupted and the population will evolve. There are 5 conditions required to maintain genetic equilibrium from generation to generation, such as there must be random mating, the population must be very large, no movement into or out of the population, no mutation, and no natural selections. The first condition, which is there must be random mating, states that all members of the population must have an equal opportunity to produce offspring. Random mating ensures that each individual has an equal chance of passing on its alleles to offspring. In natural populations, however, mating is rarely completely random. Many species, including lions and wolves, select mates based on a particular heritable traits, such as size or strength. Such non-random mating means that the genes for those traits are not in equilibrium but under strong selection pressure. The second condition states that the population must be very large. So a large population size is also important in maintaining genetic equilibrium. That is because genetic drift has less effect on large populations such as the population of birds than small ones. No movement into or out of the population. Because individuals may bring new alleles into a population, there must be no movement of individuals into or out of a population. In genetic terms, the population's gene pool must be kept together and kept separate from the gene pools of other populations. No mutations. If genes mutate from one form into another, new alleles may be introduced into the population and allele frequencies will change. Then the last one, natural selections. All the genotypes in the population must have equal probabilities of survival and reproduction. No phenotype can have a selective advantage over another. In other words, there can be no natural selection operating on the population. So we are done for the 5 conditions required to maintain genetic equilibrium. So let us proceed to the selected topics on biogeography and distribution. Remember that many parts of the biological puzzle that Darwin saw on his Beagle voyage involved living organisms. After Darwin discovered that those little brown birds he collected in Galapagos were all finches, he began to wonder how they came to be similar, yet distinctly different from one another. Each species was slightly different from one another. They were also slightly different from the most similar species on the mainland of South America. Could the island birds have changed over time as populations in different places adapted to different local environments? Darwin struggled with this question for a long time. He finally decided that all these birds could have descended with modification from a common mainland ancestor. There were other parts to the living puzzle as well. Remember that Darwin found entirely different species of animals on the continents of South America and Australia. Yet, when he liked its similar environments on those continents, he sometimes saw different animals that have similar anatomies and behaviors. Darwin's theory of descent with modification made scientific sense of this part of the puzzle as well. Species now living on different continents, such as beaver and muscat in North America, capybara and kobe in South America, has each descended from different ancestors. However, because some animals on each continent were living under similar ecological conditions, they were exposed to similar pressure of natural selection. Because of the similar selection pressures, different animals ended up evolving certain striking features in common. How biogeography considers species' geographical locations? Well, geographical barriers like mountains and oceans greatly influence the origin of species. It is therefore not surprising that the studies of geography and biology overlap in one field. Biogeography is the study of distribution of species across the planet. Let us discuss the theory of plate tectonics, which explains Earth's shifting continents. Earth's geological history has been extremely eventful. Fossils tell the story of ancient sea floors rose all the way to the Earth's ceiling, the Himalayan mountains. Littering the mountains of Nepal are countless fossilized ammonites. How did fossils of marine animals end up more than 3,600 meters above sea level? The answer is that Earth's continents are in motion, an idea called continent drift. According to the theory of plate tectonics, Earth's surface consists of several rigid layers, called tectonic plates, that move in response to forces acting deep within the planet. In some places where plates come together, one plate dives beneath another, forming a deep trench. In other areas, mountain ranges form as the plates become wrinkled and distorted. Long ago, the plate that carries the Indian subcontinent moved slowly north and collided with the Eurasian plate. The mighty Himalayas, once an ancient seafloor, rose at the boundary, lifting the marine fossils toward the sky. Meanwhile, at areas where plates move apart, molten rock seeps to Earth's surface and forms a new plate at the seafloor. As a result, oceans now separate continents that were once joined. This slow-motion dance of the continental plates has dramatically affected life's history as oceans shifted, land bridges formed and disappeared, and mountains ranges emerged. It may seem hard to imagine the Earth's continents have not always been located where they are now, but a wealth of evidence, including the distribution of some cave fossils, indicates that the continents were once united. Deep sea probes that measure seafloor spreading, along with the locations of the world's earthquake-prone and volcanic hotspots, reveal that continents continue to move today. So how species distribution reveal evolutionary events? Well, biogeographical studies have shed light on past evolutionary events, so I will answer these questions by giving two examples: the rise and fall of the marsupials and the Wallace line. Marsupials are pouched mammals such as kangaroo, koalas, and sugar gliders. The newborns are tiny, hairless, blind, and helpless. As soon as they are born, they crawl along the mother's fur into tiny milk-secreting nipples inside her pouch. Marsupials were once the most widespread land mammals on Earth, by about 110 MYA. However, a second group, the placental mammals, had evolved. The young placental mammals develop within the female's body, nourished in the uterus by the placenta. Baby placental mammals are born more fully developed than our marsupials, giving them a better chance of survival after birth. Because of this reproductive advantage, placental mammals soon displaced marsupials on most continents, including North America. Nevertheless, fossils evidence suggests that marsupials were diverse and abundant in South America until about 1 or 2 MYA, long after their counterparts on most other continents had disappeared. The reason is that water separated South America from North America until about 3 MYA, but sediments eroding from both continents eventually created a new land bridge that permitted migration between North and South America. The resulting invasion of placental mammals spelled extension for most South American marsupials. Australia's marsupials remained isolated from competition with placental mammals for much longer. Australia separated from the other continents about 60 or 70 MYA. In fact, Australia remains unique in that most of its native mammals are marsupials. Biogeography figured prominently in the early history of evolutionary thought. Alfred Russel Wallace, the British naturalist who independently discovered natural selection along with Charles Darwin, had noticed unique assemblages of birds and mammals on either side of an imaginary line in the Malay archipelago. The explanation for what came to be called Wallace Line turned out to be a deep water trench that separated the islands even as sea levels rose and fell. The watery barrier prevented the migration of most species, so evolution produced a unique variety of organisms on each side of Wallace Line. So we are done, but I would like to end up my discussion by reiterating the some important lines. It is useful to review, analyze, and critique the strengths and weaknesses of genetic mechanisms and evolutionary processes, and the evolutionary theory as well. Darwin made bold assumptions about heritable variation, the age of the Earth, and the relationship among organisms. New data from genetics, physics, and biochemistry could have proven him wrong on many counts. But they did not. Scientific evidence supports the theory that living species descended with modification from common ancestors that lived in the ancient past. The Grant research, for example, clearly shows the effects of directional selection in nature. The Grant's data also show how competition and climate change affect natural selection. The work does have limitations. For example, while the Grants observed changes in the size of the finch's beaks, they did not observe the formation of a new species. Such limitations, however, do not weaken or contradict the fact that Grant's data supports Darwin's ideas. So, is evolution a fact or theory? It is both. Millions of fossils show that life has existed for over 3 billion years and that organism has changed over time. In this sense, it is a fact, as solid as any scientific fact, that life on Earth has evolved or changed through time. Now recall that a scientific theory is a well-tested explanation that accounts for a broad range of observations. Evolutionary theory fits this definition. New discoveries continue to support new hypotheses that refine and expand Darwin's original ideas. Now scientists suggest that all evolutionary processes are fully understood. Many unanswered question remains. Why are understanding genetics mechanisms as evolutionary processes important? Because evolution continues today, driving changes in the living world, such as drug resistance in bacteria and viruses, and pesticides resistance in insects. Evolutionary theory helps us understand and respond to these changes in ways that improve human life. That's all, guys. Thank you for listening and watching. This is Norayda A.Salih. Have a blessed day, everyone, and stay safe.